Good afternoon. My name is William G, and I'll be presenting to you a portion of the paper entitled Green Tea Catechin Dramatically Promotes RNAi Mediated by Low Molecular Weight Polymers. And here are the authors. So basically, the researchers wanted to find out how to effectively transport the siRNAs within the cell by using low molecular weight polymers. First of all, what is siRNA? It is short for small interfering RNA, and it is relatively large and it is negatively charged just like DNA. Since it's negatively charged, it's susceptible to nuclease degradation. And small interfering RNA functions to regulate target genes, basically usually to turn those genes off. And it is done by siRNA RISC complex. So as you see in the figure on the right, double-stranded RNA or hairpin mRNA yields the production of siRNA duplex, and that is done by a protein called dicer. Dicer comes in and dices or cuts out some sections of the double-stranded RNA or the hairpin RNA and forms this siRNA duplex, which is around 23 base pairs long. Once that's completed, several proteins comes in and forms the siRNA RISC complex, and this is the complex that binds to the complementary RNA sequence and turns genes off. In modern world, siRNA is used in therapeutic treatment. In order to be used in therapeutic treatment, it has to be effective both in transformation or transfection to the cell and localization within the cell, and also it has to be safe for the cell or the body to use. As I mentioned earlier, siRNAs are prone to degradation by nucleases, and once it's degraded, then there will be no therapeutic effect. So in order to prevent degradation, researchers came up with several methods, but in this specific paper they talked about cationic polymers. As seen in the figure below, the free siRNAs, which are negatively charged, associate with cationic polymers, which are positively charged. And what these cationic polymers basically do is they surround the negatively charged siRNAs to basically coat the siRNA so that nuclease won't be able to degrade the siRNAs. And this complex forms a siRNA polymer nanoparticle. What they find out by trying many different types and different weights of polymers is that when high molecular weight of polymers are used, they yield high efficiency. However, they also yield low cell viability. As you can see in this diagram here, the blue figures are polymers with relatively high molecular weight. They yield higher percentage of transfection efficiency. However, most of the cells die. However, when we use low molecular weight polymers, despite yielding low efficiency, a lot of cells survive. As you see, the red figures over here, they are not pretty efficient. However, most of the cells still survive. So what scientists wanted to find out was what kind of molecule yields high efficiency yet also yields high cell viability. And what they found out was that this molecule called EGCG, short for epigallocatechin 3O gallate, yields high efficiency along with high cell viability. EGCG is a major ingredient of green tea, and as you see in this figure, a lot of phenyl groups or phenolic groups, they form hydrophobic and hydrogen bonds with the DNA, or in this case siRNA, and it forms a negatively charged complex. As you see in this figure over here, siRNA along with EGCG forms a complex which is negatively charged. And different types of cationic polymers associate with this negative complex to coat around the siRNA or the complex to prevent from degradation. Now, they knew that EGCG associates with siRNAs, but they wanted to confirm that EGCG and siRNA actually interact with each other. And they used ethidium bromide to do this. Ethidium bromide is a substance that intercalates in between the RNA or in the grooves of RNA, and once it's intercalated, it produces a red fluorescent light. As you see in this figure, the red line denotes the light that is produced when ethidium bromide is intercalated within the siRNA. As you see, the intensity of red fluorescent light is pretty high. However, the dim gray light denotes the fluorescence that's emitted when EGCG is also present within the complex of ethidium bromide and siRNA. As you see, the gray or the dim gray significantly dropped from the red line. So basically, they concluded the addition of EGCG significantly decreases the fluorescence intensity, which concludes that EGCG actually interacts with siRNA. Now, as I mentioned earlier, siRNA alone, or free siRNA, since it's negatively charged, is very susceptible to degradation by nucleases. So they wanted to find out if EGCG and siRNA complex actually prevents degradation. So in this column over here, EGCG and both RNase, which is endonuclease, are absent. So when they're both absent, band is formed, or band is shown. Basically, that means siRNA is not degraded. However, when EGCG is absent and nuclease is present, a band is not shown, which indicates that the siRNA was degraded. However, in these three columns, EGCG is present and endonucleases are also present. The results indicate that the siRNAs are not degraded, even though the concentration of the endonuclease or the nucleus increases. So they could conclude that EGCG siRNA complex prevents degradation by nucleases. They also found out that the complex is negatively charged. They did not describe in detail or what kind of method they used to find this out, but the results basically says that EGCG siRNA complex is negatively charged. The next step, or the next procedure that they used, was isothermal titration calorimetry. So this specific procedure measures enthalpy and entropy of the biological reactions that we're looking into. And this is done by sensing minute changes of heat released or absorbed within the apparatus by sensitive thermal sensors. So as you see in this apparatus over here, there are two cells, one as a reference cell and a sample cell. And both of these cells are in a isolated apparatus, meaning heat is not released or absorbed into and out of the system. The reference cell has water or either a buffer as a control, and the sample cell actually has the biomolecules that we needed to react to see the entropy and enthalpy changes. So what they found out through this isothermal titration calorimetry, as you see over here, there are upward peaks, which means the reaction absorbs energy from the surrounding. So they concluded that EGCG interaction with siRNA is endothermic. So from this short section of this paper, they found out that EGCG siRNA complex is not degraded by endonucleases or nucleases, and also that EGCG forms negatively charged complex with siRNA, and that EGCG siRNA complex, or the negative complex that formed, associates with low molecular weight cationic polymers. As you see in this diagram over here, negatively charged siRNA associates with EGCG, forming a negatively charged complex, which allows interaction with low molecular weight cationic polymers, and those cationic polymers forms a coat or shell around it to help them transport into the cell and also prevents them from degradation. And once it forms associations with cationic polymers, they found out that it results in high efficiency with high cell viability as well. So for future research, they have to try or experiment this EGCG siRNA complex in clinical settings, basically to humans. They proved that it works in laboratory, but now they have to find out if it works in humans. And they also would be beneficial if they found out more substances like EGCG so that we can have more options for different types of treatment. And here are my references. Thank you.